Thank you for being here and for your time and attention this week. It's really important that we continue to collaborate and discuss these issues. With that, it's my pleasure to introduce Janet Lambert, our wonderful CEO, who's going to tell you a little bit more about the work that ARM's doing today. So thank you very much.
Thanks. Thanks very much, Matt. Yesterday, for those who were here in the opening remarks, I had a chance to tell you a little bit about ARM and what we're doing, and today I'd like to expand on that and give you a little bit of a greater flavor for the efforts that we have underway on behalf of the sector as a whole. So for those who might be new to ARM, ARM is an international advocacy organization. As Matt said, we're dedicated to bringing safe and effective regenerative medicines to patients around the world. We do that with the help of our members. We have almost 350 members at this point. They are diverse and include nonprofits as well as commercial firms, and they span 25 countries now, which is wonderful. So our priorities have been constant for many years, and as Matt said, there's a lot of issues that are standing between the great scientific work that's happening in this field and patients after actually receiving them. So those things include regulatory pathways, reimbursement policies. We have a number of manufacturing issues in this sector, as well as just the need to communicate both the promise of these therapies, the state of the field, and the challenges we face. And then lastly, we have a very significant number of small companies who are still in the fundraising part of their journey, and so we try to facilitate their access to capital. So to dive into these things just a little bit more, the number one priority on our radar screen this year and next year, I believe also, is to drive the adoption of new payment models and value-based reimbursement. As I think most of the folks in this room know, the therapies we're developing don't fit into the standard model of a pill a day for 5, 10, 15 years. So we're trying to affect those rules that limit the adoption of more creative payment models, such as the ability for someone to receive a curative treatment, a one-time treatment, but to pay for it over time, or for therapeutic developers to enter into what we call pay-per-performance or outcomes-based agreements with payers that link the payment they will receive to how well the patient is doing on the therapy provided. Those things are actually highly limited by a set of rules, price reporting rules in the governments. There was some conversation about this in a panel yesterday that you may have heard. And so what ARM does is lobby the Congress about those rules. We deal with the administration, the Congress, other stakeholders to try to get those specific rules changed to enable the field to move forward. Likewise, there's a conversation that we undertake with CMS, the Centers for Medicare and Medicaid Services, about a specific set of reimbursement rules, a DRG, NTAP, IPPS, for those of you who are in this world of detail. But for those of you who aren't, it's just a certain set of reimbursement rules that the government sets, which currently are making it cost prohibitive for many hospitals to provide CAR-T, despite the remarkable clinical effect that CAR-T is having on patients. In addition to doing that kind of direct lobbying and direct engagement on behalf of the sector, we do some analytical work to support that. So we'll produce analyses of what's the current state of play in Europe with respect to market access for ATMPs. How does one country compare to another? And we use that to then talk to policymakers to try to get them to come up to the highest standard. The other thing we're doing a lot of work on and have for some time are regulatory pathways, and we're really excited that Peter Marks from the FDA is here today and will be able to speak about the activities underway in the agency. But ARM was deeply involved at the FDA in the creation of the RMAT designation, the expedited approval pathway for regenerative medicines, and ensuring that it included both cell therapy and gene therapy. We continue that dialogue with the FDA Now much of that dialogue is focused on CMC and manufacturing guidance-related issues. At the same time, we have that same kind of interaction with Guido Rossi and his team at the European Medicines Agency, and I've had really a terrific year of working with them on issues that are important to the sector and how the EMA can help us both at the pan-European level and at the country-specific level to get ATMPs to patients. Thanks to those conversations, we've also been invited to give a talk at an international coalition of medicines regulators that occurs twice a year, I believe. And we've been asked to come and talk to this gathering of the world's medicines regulators about what it is that the ATMP or regenerative medicine sector needs from those global regulators in order to get our products to patients. On the manufacturing front, that's less of a public policy issue. We're not lobbying Congress or the administration to change rules so much as we're trying to convene the community to understand what are our collective challenges and barriers, and what are the things on a pre-competitive basis that we can do together. So this chart shows a list of companies that have come together to work on a project called A-Gene, which is, for those of you who know the monoclonal antibody world, patterned after a project called AMAB, and is really an effort to bring together best practices, a book of knowledge, we sometimes call it, about gene therapy manufacturing, because we know the field is growing fast, many people are coming to it anew, and there isn't really a cookbook per se of how it is one should go about this. And this is another project where we're in touch with the FDA about it, so that there's also some regulatory kind of how-tos embedded in this. We're completing that project on the gene side, and it should be out in the first quarter, and we've already launched the cell version of that. In addition, we'll pull together the community to tackle specific issues that have come up in our conversations with members. For example, comparability, we know, is a key issue in manufacturing and the regulation of manufacturing in this space. So we brought a bunch of our member companies together in June to kind of talk about those issues, and the FDA and the EMA participated in that dialogue to make sure we had their point of view and they had ours. In this vein, we also launched, and I wanted to make sure and note, the standards coordinating body. There are a number of standards efforts underway in this space, and the FDA actually is funding this entity, which was incubated at ARM, but is now its own independent organization, driving 15 different standards advancements projects in this field. If you're unfamiliar with it and in this field, I would recommend that you either find the SAB folks who are here or go to the website. They've produced some good documents that both summarize the state of play and the gaps in standards development in cell and gene therapy. ARM also participates in international dialogues. Most notably, there's been a lot of conversation, as you know, about germline gene editing and events in China and elsewhere. Part of the way we engage in that is we put together a statement of principles of therapeutic developers in this field. It was our point of view that the international dialogue that was occurring was largely academic-driven, scientifically driven, which is appropriate, but there is also a voice for those companies that are using gene editing in their work to describe where their emphasis is and where their emphasis isn't. And so we pulled together this statement of principles. 14 member companies of ARM signed it. We were asked to present it at the World Health Organization expert panel on gene editing. We've been asked to talk about it at the national academies and other forums, and we'll continue to kind of drive that point of view in those dialogues. We also produce a number of reports that are available both to ARM members and to the general public. Those include a global overview of the sector, regional overviews. We do indication-specific reports about what's happening in rare disease, what's happening in oncology in this sector. Those reports give you information about the number of companies working in this space, what's the status of their clinical trials, what indications are they focused on, how much investment has come into the space, and the like, and gives you the ability to track that over time. We've also deployed a couple of databases this year so that if you're interested in finding out what are the upcoming data events and clinical trial results, You can do that through a easily searchable database on the ARM website. And we also have a list of those therapies that have received expedited approval pathways, either PRIME or the expedited approval in Japan or the United States. Finally, we produce a couple of newsletters that folks find valuable. One is a sector newsletter where weekly we cover all the developments of the sector, policy, finance, clinical, and the like. And then we do a specific monthly briefing for our members that talks a little bit more specifically about what is the work we're doing, what are the committees that ARM has and does its work through focused on, and the like. So please take advantage of these if you would like to. They're there for your purposes and your help. Lastly, I want to note that ARM did launch a foundation in January of last year to focus on the vast educational needs in this space, and we felt there was a lot of work that could be done in the context of a 501c3 charity that would be additive to the field, and the leaders of the foundation will give a talk about that tomorrow morning at this time and in this room. If you're interested in learning more about it, I encourage you to attend. This is the ARM Board of Directors. You heard from Matt Patterson earlier. It's a really illustrious group of people who have been really helpful to me and the team in driving the work of ARM, and I encourage you to talk to any of them or to me if you have any questions about ARM. Thanks very much. Microphone on. I'd like to introduce the next panel, or the next panel moderator, at least. One of the things that's really thrilling about the moment in which we gather is that we're actually starting to have a lot of experience of delivering cell and gene therapies in the clinic. And we wanted to use this occasion to bring some of those folks who are on the front lines in the clinic delivering CAR-Ts, have them have a chance to come and talk to you about the particular challenges and opportunities they see. So to moderate this panel, we have Gregory Hale, Senior Medical Director of Hematology and Oncology at Medpace, and I'll invite him up, and I think you're going to introduce your panelists, so thank you. You're welcome.
Good morning and welcome to the talk entitled Clinicians Working with CAR T-Cell Therapies. This is a very fitting session currently because we now have two CAR T-cell products that are commercially available and a large number that are in clinical trials. I'm a pediatric hematologist oncologist by training, and I have spent 26 years in this field, most of it in academic medicine, being a site investigator in many cell and gene therapy studies, and I now currently work for Medpace, operationalizing clinical trials in cell and gene therapy for various sponsors. And it's certainly impressive. When I started this field, many of the trials were in gene marking, and so it's really exciting to see how we've moved the mark, and now we're able to introduce therapeutic cell and gene therapy products for patients. And so it's a really exciting time in the field. What I'll do is we're fortunate to have two speakers who who have extensive clinical experience in CAR T-cell therapies, and what I'll do is I'll allow them to introduce themselves before we start with the question and answer period.
City of Hope. And I'm an infectious disease specialist. I'm not actually a card-carrying immunotherapist, but at City of Hope, I am the institutional official for research as well as the director of the Center for Gene Therapy. So I have a basic science knowledge of this area, but also I have a clinical area because it's the fever that drives much of the complications and the management following infusion of CAR T cells. So infectious disease becomes involved with patients. So we see all the CAR patients, and we are familiar with the management strategies, et cetera. So I'll be able to talk about some of that aspect.
My name is Dimitrios. Tzachanis. I'm an associate professor at UCST here in La Jolla. I'm a hematologist oncologist by training. I'm in the bone marrow transplant division at UCST, and we're administering the commercially approved CAR T-cell products, as well as we're doing the clinical trials with CAR T-cells. And I'm the site PI for the CAR T-cell trials for lymphoma and acute lymphoblastic leukemia at UCST, and I'm the leading physician in the commercial CAR T-cell program.
Thank you. What we'll do is we'll have some question and answer and discussion initially, and then I'll open it up in the last 15 minutes to questions from the audience. So be thinking of any questions you have, and we'll address those towards the last quarter of the session. For the first question that I'll open for our panelists is just that patients who are receiving CAR T-cell therapies often have had a lot of extensive treatment. They have very bad malignancies, relapsed refractory malignancies, and therefore they're put into the pathway to receive these more aggressive therapies. What are some of the key components of a clinical CAR T-cell program that are important in differentiating an oncology program?
Let me talk about that, the way we've done it at City of Hope. City of Hope actually has had a CAR T-cell program for many years, and we, in fact, when we recruited Michael Jensen 20 years ago is when the program really started. And to think about how to create a program, you need commitment, of course, at the top, because you're certainly going to need facilities and you're going to need resource allocations. So you need a governance plan. And our board of directors had a strategic initiative based around CAR T cells that they established probably 6 or 7 years ago. So with that, we have developed a governance plan which really has like 4 pillars in it for each of the major areas. There is administration and the issue around facilities. There is the area of just administering the process fiscally, billing and that sort of thing. There's the clinical aspect, that is, who screens these patients and who says that they can have a commercial product that the institution is going to commit to financially. And then finally, the research pillars. So that's the top-down look at it, and we have expert people in those areas. There's also the problem that you allude to. These are very sick patients. These are the sickest of patients that we have. These are the patients that are dying of hematologic malignancies, and they need tremendous amounts of support. So you can imagine a medical center that might start a asthma center. They know they have some kind of support that they need, but it's relatively discrete and defined, pulmonary function studies and that sort of thing. But when you do a program like this, you need expertise across the board, because the complications you're going to see are going to be systemic, certainly neurological, certainly pulmonary, certainly liver, renal, and of course hematological. They'll be neutropenic. They'll be B-cell deficient for years. So you need that kind of expertise then at let's call it the bottom-up phase. And so you need to have people that are, let's say, trained. So not all of our people are allowed to use CAR T cells. You must be certified. And again, it's certified by this governance body, especially the committee we call the Clinical Cancer Immunotherapy Committee, the CCIC. So there you talk about REMS, which is risk, evaluation, and mitigation strategy. If you understand that for managing a patient, then whether you're the nurse, whether you're the clinical research coordinator on the trial, or the clinical research nurse on the trial, or the physician managing the patients, you'll all have one approach, and you'll be following the various let's call them mitigation strategies, which are really SOPs. So we have 10, 15 SOPs. I reviewed them in preparation for this, and I was impressed with every aspect of patient management is covered by an SOP. And all of these are derived from publicly available documents. It's not like it's special to our center or UCSD or any other center. Your center can easily put together the documents you need so that people in your center are well qualified. So you have the top down and you have the bottom up type of activities around this patient. Now, the families, of course, need a lot of support. We have for many years developed a patient education center, which is really right in the center as you enter City of Hope. And the families can go in there and be educated. They can we have a computer section for them if they want to just look up stuff. We have a little teaching area with an auditorium, not an auditorium, but a classroom where we give sessions for the families. Because they must understand, again, the complications. And we talk about having them understand the package insert. That is the risks and things that are actually associated with the commercially available products. So, and there the nurses get involved a lot. And it's really the nurses that make the difference in any program. And that's a key aspect, I think. So I think I'll stop there and go on.
And so when you look at clinical programs, many of these patients do cross treatment services. It's not uncommon they end up in the intensive care unit. Some of their toxicities are neurologic, so you need a neurologist. How do you integrate all the different aspects of the clinical services of a hospital to sort of know how to intervene with these patients? And I guess the second question in that is, for your CAR T-cell therapies at your institutions, are they administered entirely inpatient? Do they start outpatient? Or are they a combination of both?
So to answer the first question, I think we're more of a moderate-sized program compared to City of Hope. They're a bigger program. So we relied heavily on the experience of the bone marrow transplant program because a lot of these things like the patient education, taking care of sick patients, the FACT accreditation, the SOPs, they're very similar to what we use for our allogeneic transplant patients. So we relied heavily on that. But we also had to take into account the unique side effects that we see with CAR T cells. And as you alluded to, they're mainly, these side effects are mainly co-managed with our ICU colleagues and our neurology colleagues. So a lot of the unique effort that went into building the CAR T cell program had to do with educating our colleagues from the ICU and from neurology. We had dedicated persons in the ICU and neurology that helped us write the SOPs together and our protocols, how we're going to triage somebody sick. And we have now certain pathways in conjunction with neurology and ICU. So for instance, when we admit somebody for CAR T, we already work together with the ICU so that there is a bed available for them. And we have designated providers in neurology who will see those patients if they develop any neurologic symptoms, and not just a general neurology consult that wouldn't work. And what was the second part?
And so
of the question
at your
about
center, is the lymphodepletion, which is a chemotherapy
Yes.
regimen that's administered prior to the infusion of CAR T cells, administered inpatient or outpatient?
So the lymphodepleting chemotherapy we started administering outpatient at this point, but we do admit patients by day 0. So all CAR T cell infusions are being done inpatient.
Okay. One of the issues that always comes up at least in clinical trial aspects and also clinically, is With autologous CAR T cells, there's that initial collection period, generally by apheresis, and then the CAR T cell product has to be manufactured. In that circumstance, how often, in your experience, does a patient donate and is then unable to receive the CAR T cell product because of some clinical circumstance, because of the time it takes to manufacture? And do you have a certain standard or practice in the use of bridging chemotherapy in that circumstance?
So that's a very good question. And I would add to that there is also a period from the identification of the patient until we can get to apheresis, which is something that we didn't have to deal with in clinical trials, but with a commercial product, we have to get approval by insurance, we have to do all the workup, and that can take another 2 to 3 weeks at least. So we have to add that period before the leukapheresis. And these patients have aggressive disease. By definition, they're refractory to chemotherapy, and they might have a lot of side effects from prior treatments as well. So we don't have a standard of care bridging therapy that we go to. It's very patient individualized. A lot of it depends on what they've received before, if their disease is still responding to steroids, because we try to start with just steroids. And we try to avoid any chemotherapy, such as purine analogs that can wipe out T cells for a long time, because we don't know what the effect of that will be. And so far, we haven't had any difficulties getting some patients from leukapheresis to CAR T. We've had one patient on a clinical trial with, who was heavily pretreated with ALL, and the cells didn't grow. So we had one failure, but so far everybody else we've been able to get to CAR T one way or the other.
Okay. And once you
Let me
infuse...
just say that historically, of course, that's been a problem. But as the processes in manufacturing get better, but remember, there were times when the Kymriah product did not always produce a product because you're taking a patient's, very recent patient that may have 70% or 80% blasts in their peripheral blood. And we require that you have at least 100 CD3 cells before you're eligible. But that's still a challenge to get out those cells and to get them selected and genetically modified. That, I'm told, is a problem that is going away, that is the problem of production. But then you also have the problem of the patient has to survive while they're waiting for this. And that's always a problem. And up to 20% of patients can expire while they're waiting for the product to arrive. So that's another issue. The issue of the product that is made but doesn't pass release tests is another issue. And so there's actually a trial now at our center and other centers where you're allowed to release the product that fails specifications. And the patient leaves the area of a clinical product and joins a clinical trial and agrees to use those cells that did not pass specifications but are CAR T cells and they're functional. So that is another way to solve the problem of getting the product to the patient.
Especially for this patient population. Mm-hmm. So we've talked about a lot of the issues with autologous CAR T cells, and that sort of brings up a lot of the newer products, maybe off the shelf or allogeneic CAR T cell therapy. So in that circumstance, do you see do you have any experience with allogeneic CAR T cell therapies, and what do you see as the big advantages over autologous CAR T cell products, as well as disadvantages?
We have active the Precision Bio trial at City of Hope, and it's active at other centers around the country. That is an off-the-shelf product in which the T cell receptor alpha chain is knocked out of the CARs, and so that the CARs will no longer attack the recipient. That has enrolled a number of patients. I've been told that they're going to present their results in the first quarter of 2020. And that is obviously one of the goals, I think, of the field, is to have off the shelf. And then you eliminate that problem of waiting several weeks or months before you can get the product, and while the patient's very sick, possibly in the ICU, generating tremendous amounts of cost just to maintain the patient while they're waiting for the cells. So when there's off-the-shelf CARs, this is going to change the whole field. It's also going to drop the cost dramatically.
And it might work better in theory because you're taking lymphocytes that are much healthier than the lymphocytes from the patient who have seen a lot of prior therapies.
Possibly.
Yeah, that's a good point. In my role, I'm always looking for CAR T-cell programs that would be good fits for various CAR T-cell trials. As a site, what are the aspects of a clinical trial that you look for when you're trying to determine whether or not you want to participate in a trial? And what, and the second question is, when you decide on a trial, generally, what is your time frame and pathway to getting that trial open? Because time's always an issue in opening trials.
We have again, in the governance system, we have a very strict approach to what new trials do we begin. We have an SVP, Ashley Baker-Lee, who may be here, you may know her. But she set up a system of disease teams where the physicians and the scientific staff are together, and they must commit to a trial. They must endorse the trial. So they look at their portfolio and they look at their patient population and they say, we can do that trial or we can't do it. And so there's a number of disease teams, but one of them is the immunotherapy disease team. So that is committee that a physician or an investigator would bring a new trial and say, we think there's a window of opportunity for this trial, we can do it quickly, we've got the patients, et cetera. And then they will endorse it. And then at that point, the clock starts running, and they must get it approved within so many weeks and get that first patient enrolled. So that's the approach that we take.
We take a similar approach. We also have our disease teams where we discuss new potential clinical trials, and we always look, number one, at unmet clinical needs. So do we have any overlapping trials? Is this a patient population that really needs some treatment? Like, for instance, now patient failures after prior CAR T is a big unmet medical need, so we're always looking for trials in that area. And number two, innovation. How cool is this new idea? What can bring to clinical practice? For instance, the off-the-shelf products are extremely interesting right now because of the issues we mentioned before. And then from start to from the very beginning until we're able to put a first patient on, I think we need at least 6 months, which is always a problem, but it has to go, especially for these new products that have to do with cells or genes. They always have to go through special committees. The IRB is not so much an issue, is getting to the IRB.
Mm-hmm. Right, yeah, usually there's a little bit more intense scrutiny from institutional regulations and regulatory committees as well as the budgeting aspects.
Exactly.
We did have talk a little bit earlier about the toxicities. We know that patients who undergo CAR T-cell therapy, at least with the commercially available brands, have a relatively high incidence of cytokine release syndrome, as well as neurotoxicity. And so in your center, one of the things that comes up in clinical trials is the guidelines about when to intervene with treatment, which is usually tocilizumab and or steroids. And does your program have defined SOPs or criteria that sort of standardize when to intervene, like a certain toxicity grade of the cytokine release syndrome or neurotoxicity, or do you sort of leave that up to the medical decision-making of the physician on service?
I can start by telling you, it's a very strict protocol and SOP that we have. So when you think of evaluating the patient, you're thinking about, usually it's initiated, the concern is initiated by fever. So if the patient's above 101 degrees, that's the first signs that you're going to have a cytokine release syndrome. And then you combine that with whether or not there's hypoxia or not, and you can begin to grade the patient. you're actually looking at neurological signs. You ask the patient, there's a scoring system in which there's a 10-point scoring system in which you ask the patient some questions. You ask them, where they are, time, place, location, what hospital you're in. Then you ask them to do something. You ask them to recognize, your fountain pen. You ask them to write. And so the patient can make 10 points. And if he only makes 1 or 2 points, he is, a grade 3 neural toxicity. And so that's a major problem. If he's at 9, he's got a fever, but he's not having a major problem. We don't treat unless it's a grade 2 for 3 days, or if it gets grade 3. At grade 3, we admit them to the ICU and we start treatment. And at that point, they may progress very rapidly or they may not. They may begin to resolve.
So to answer your question, we do have very detailed SOPs, and we do have very strict monitoring guidelines, like the neuro assessment that Dr. Zaia just described. But I have to say, from the practical standpoint, it's never clear cut. They don't always fit a box. And we also have to remember that these are all guidelines. There's not much data supporting every little decision that's in those guidelines. So all these tools are supposed to help you make the decision. They're not absolutely strict. And practically speaking, a lot of times we have to make a decision quickly, and we might deviate a little bit from our SOP because the patient might have symptoms that are related to the underlying malignancy. They might have symptoms from other comorbidities. They might have symptoms from the lymphodepleting chemotherapy, and it's not always easy to decide if the headache that they have is from the Zofran that we gave them, or if they're starting to develop a CRS, or is it the first sign of neurotoxicity. So we try to follow the SOP as closely as we can, but it's not always that straightforward. And we, like I said, we have a team, a small team of experienced clinicians from many specialties, neurology, ICU, and a lot of times we put our heads together to decide when we pull the trigger to start the steroids or the tocilizumab.
Yeah, and I think you make a good point that all these patients are quite ill and therefore very challenging. And when you have this many comorbidities, the differential diagnosis of what can cause a problem, can be quite long. I think the second point that's important that you made that I just wanted to emphasize was that it's having a core group of specialists who have expertise, like a neurologist or an infectious disease doctor who are familiar with CAR T-cell therapies and their complications, because you can kind of build on that experience rather than having, the less experienced specialists involved. One thing that John had pointed out was about fever being the first symptom of cytokine release syndrome, or a lot of complications in these patients. For patients who receive CAR T-cell products after a lymphodepleting regimen, do you recommend any, do you do any special antiviral or antifungal prophylaxis at your centers? Do you do certain viral monitoring, like for viral reactivation, such as after allogeneic transplant, or what's your infectious disease monitoring and prophylaxis like?
So we prophylax for fungal. These patients are basically hematological patients at high risk for whatever is associated with their disease. So if they're an ALL patient that's failed multiple cycles and have been neutropenic for weeks, they have all the same risk factors that you would normally be worried about. So you'd have the patient on antifungal coverage, prophylactic coverage, and you would have them on, let's say, cyclovir or some prophylactic for herpes simplex and for pneumocystis. Given that, we know that there's going to be maybe 40% during the cytokine release syndrome that will develop a bacteremia, maybe not 40%, but 25 to 40% based on the published results. And the question is, should you prophylactically give antibiotics? And we've chosen not to do that, but we then treat empirically when they have the fever. So we would begin the antibiotics on the first day of the fever. And I'm not sure if other centers use prophylactic antibiotics, but I think you're asking for resistant organisms if you do. We tend to be pretty conservative about the use of antibiotics to prevent the problem of resistance. So that's basically the way we manage the infectious disease aspects.
Okay.
So similarly, everybody's on acyclovir and PCP prophylaxis. And for at least 3 months, because they also receive they've also received a purine analog before as part of the lymphodepleting therapy. We do use fluoroquinolone for prophylaxis, so we're more liberal when they're neutropenic, when they're absolutely neutropenic. And lately, we've seen some CMV reactivations, so we started checking CMV viral loads weekly, similarly to what we do with our allogeneic stem cell transplant patients.
Okay. And in the CD19 targeted autologous CAR T cells, hypogammaglobulinemia has been, reported to be a significant occurrence. And I don't know how that, what you've observed in practice at your institution, and how does that translate into protracted management? Do these patients just get monitored? Do you replace with IVIG, or how do you follow up the adult patients that you see with this complication?
So we monitor them just like you would monitor a patient post autologous transplant with looking at the serum immunoglobulin. So if it's at 400, we don't give supplemental immunoglobulin. But these patients will most likely, or many of them will be below 400, and you will give them periodic immunoglobulin. Because remember, the CD19 knocks out the B cells, and so the B cells make immunoglobulin.
Yeah, everybody will become hypogammaglobulinemic. The question is who needs the replacement with IVIG, and I don't think we have a good answer to that. We definitely give IVIG to everyone who has had infections, especially bacterial infections. We don't have a strict policy about giving IVIG to patients who just have a low IgG, but no history of infections. But we're looking at it.
And one thing I just wanted to go back to follow up on was when you care for the patients on the inpatient service, or do you care for your CAR T-cell patients on the transplant clinical service, or on the quote-unquote leukemia or hematologic malignancy service in the case that they're separate at your institutions?
So that's a better question for John, because we only have one service.
One service there, okay.
So everybody's on the same service, whether they have an allotransplant or CAR T or being induced from leukemia.
A CAR T-cell service.
Okay. And then the one other aspect, because they are gene therapy products, everyone's always wanting to comply with the regulatory requirements for long-term follow-up. And so, which is always, for this patient group, is always a great problem to have, but how do you follow up the patients who've received the gene therapy product? Do you have like an internal long-term follow-up trial that they sort of enroll in for follow-up, or how do you meet those requirements?
So if you look at the problem, in fact, there's two publications that have you might want to refer to. One is by Josh Hale from the Fred Hutch, and he published this in Blood in January of 2018. And then Park summarized the Sloan Kettering results for infections. And there's two kinds of infections, two waves, let's call it. You have the immediate early problem in the first month, and then you have the long-term problem. So you're referring to the long-term problem now. And there it's like any patient who is, let's say, call them immunosuppressed or post transplant. I mean, these people will often, some of them will be neutropenic for many weeks, and so they're at risk for infection. But most of the infections that occur now are fungal or viral. And the leading viral infection in the Seattle experience was community respiratory viruses. So they're going to pick up whatever is in the community. And you worry about RSV, respiratory syncytial virus. Certainly you worry about influenza. These people are not going to be able to respond to an influenza vaccine if they've had a CD19 treatment. So you may have them on fungal prophylaxis longer than usual. So that's the main problems late. The early problems, in terms of follow-up, we follow them for the first 4 weeks very closely and seeing them at least, our SOP actually says at least once a week for 4 weeks. We actually see them more frequently than that because they're usually in-house and as inpatients, and we see them every day. But once they're discharged, then we see them weekly. And we would monitor them for, the signs of some late, you can actually get a late cytokine release syndrome, so we monitor them with these acute phase reactants, the so-called C-reactive protein. Ferritin is another acute phase reactant. And we looked for, just the usual CBC and metabolic panel, as we call it. So that's basically the way we follow them.
So in terms of long-term follow-up, I mean, we hope that, a great proportion of these patients will be cured of their disease, and that's what the data suggests so far. We haven't treated enough patients to have a specific long-term follow-up clinic for CAR T-cell patients, but we do have a long-term follow-up clinic and guidelines for lymphoma survivors. And that's what we follow for the patients who get to, let's say, a year after CAR T-cells.
And what's your criteria for discharging them back to the referring oncologist?
That's a good question. I don't think we have strict criteria because a lot of times, practically speaking, this is driven by insurance and not by our decision. But generally speaking, we like to follow at least the 100-day rule, at least the day 100. Because as John mentioned, a lot of these patients will develop problems, especially infectious problems, even after day 100. So ideally, we'd like to see them after that too, but at least until day 100, we try to keep them at our center.
Okay. For patients who have an episode of severe neurotoxicity and recover from that, do you see any long-term sequelae in patients once they are treated?
I haven't seen any. It might take a long time for every symptom to resolve. Like they might still have a slight tremor for months after their episode. Or something of that nature, but I haven't seen any long-term sequelae for just pure neurotoxicity.
Okay. Earlier, I think John made a reference to the financial aspects, and yesterday there was a session talking a lot about the financial costs of cell and gene therapies. How has having a CAR T-cell program at each of your institutions affected them financially, and is there an impact on patient access to care, or is that something that you've managed to develop a process that runs smoothly?
You want to answer that? I mean, it's a real problem. I mean, the institution loses money on most commercial products that we use. It has been estimated. We know that from Medicare that because they have the NTAP, this is the new technologies, added payments, last spring they up to 65% of the acquisition costs, which means you're still fronting 30% of the cost. So it's estimated to be about 130 to 150 or so that an institution will lose just there. But of course, you're managing the patients as well during the period, and as they approach being very sick, and they may be in the ICU while they're waiting for the therapy. It's been estimated, there's a study out of Oregon that was reported, Oregon Health Science Center, that was reported at UPenn at one of their conferences this year, where it's estimated that the cost may be up to $1.5 million for the management of a patient. And when you look at other similar cancer therapies, if you take a lymphoma patient with diffuse large cell lymphoma who's failed 3 and 4 cycles of therapy, and you're managing them with these 3 and 4 cycles, it's estimated that costs about $750,000 per year for that patient, and the outcome is dismal. The CR rate is 7%. The complete remission rate is only 7%. So that you're getting probably a more value for your dollar if you use CARs, but you're still, you're driving that Maserati, but you still can't afford it. That's the basic problem that we have, and that's that is the issue that we're discussing, I think, in many areas, and it's the topic of discussion here at this meeting. It's got to be solved because it's not a sustainable model. Institutions are not going to continue to lose money. I can't imagine how a state, we actually have ways to bring in donor dollars to support this, but I think a state institution may be more limited. I just don't know.
Well, we have similar problems because a lot of our patients have Medicare or medical, so we don't. We lose money. I think it requires strong commitment at the very top of the institution from the academic centers who now say, okay, we were losing money, but we want to be part of this. We cannot afford to stay behind the wagon. But I don't know for how long it will be sustainable. So I think we need to lobby more to try to find a solution to this problem because it is a real problem. I know that in our institution, our program is under a lot of scrutiny at the very top. So they're looking at the finances on a monthly basis and see how much money they're losing. And at any given time, they might pull the plug. They might say, this is too much of a loss for our institution.
Do you find that when patients are referred, prior to apheresis and the cell collection, you had mentioned earlier it may take 2 to 3 weeks for the insurance negotiations and authorization. Is that, are there time periods where that's longer or it impacts patient care?
Yes, there's been time periods that are much longer than this. First patient that the VA referred to us took 2 to 3 months until we were able to sign a contract with the VA and move forward. Thank God we were able to control his disease in those 2 to 3 months, but if there was somebody else, maybe we wouldn't have been able to do that. That's also a big issue.
I guess we'll move on to one of the factors that we talked about a little bit earlier was patient's perception of the treatment process. And as someone who's had transplant gene therapy a long time, I've had patients who go through transplant, and they're like, oh, that was worse than I thought it was, or it wasn't as bad. I didn't know from your experience with patients getting CAR T-cell therapies and a lot of the, there is a lot of hype around response rates, but there's also a lot of concern about toxicities. Once patients go through the consent process and then they go through the CAR T-cell procedure, do you have the sense that they think it was more difficult than they thought it was going to be, or are they disappointed in the results, or what? Do you have a sense about what the perceptions are about the whole process?
Well, it depends on whether they respond or not. I think patients who've responded and they have these amazing results, they've never complained about the process being too difficult. Even if they were in the ICU, even if they were on pressors, even if they were intubated, they find the results so gratifying, and then they don't have the cancer is gone basically. That I haven't had anyone complain, and also they don't have the long-term side effects that they have with an allogeneic transplant. Let's say they don't have to live with graft-versus-host disease. So most of the symptoms that they had before the CAR T cells were related to their malignancy, and they're gone. So they feel a lot better after this process. So even if they have to go through this relatively short period of suffering with CRS and neurotoxicity, nobody has complained to me. Nobody has come back and said, well, I wish we hadn't done that because I was in coma for 3 days.
I mean, this is a patient population that's been conditioned by often months and even years of cycles of chemotherapy. So it's not like this is a surprise. I mean, one of the biggest surprises, I think, for us is that the patient expectation changed with CAR T cells. When you fail 3 and 4 cycles of chemotherapy with an adult ALL, you're very sick, and you're talking to the patient about hospice care and end-of-life care. That conversation was curtailed in many cases. I don't know if you've seen this or not, but patients say, I'm not going to go to hospice, I'm on CAR T-cell therapy. If we can't get CAR T-cell therapy, then we can talk about it, this sort of thing. And that changes the whole dynamic of patient flow into the hospital and staying in the hospital. So it really accumulates census. So our census in the hospital has been a major problem in the last 2 years.
Do you ever get the sense that there's like the patients expect sometimes too much, or that there's over-promising? Because from what you read in the news, it's sort of like a miracle drug. I mean, I know when patients come to you, do they have a sort of an unrealistic expectation that this is like a slam dunk, or are they aware appropriately of all the potential complications that they're at risk for?
I do think that a lot of the patients, they go online, they Google CAR T cells, and they do have very high expectations. And we have to remember that the so-called cure rate is probably in the 40% range at this point. So most patients won't respond to the CAR T cells still. And also, a lot of the times, for reasons that are related to their underlying disease, they might not be able to get CAR T cells, or a transplant might be more appropriate. But because they've researched CAR T cells, they've been on Google, they've been reading all that publicity, they really try to get a treatment that might not be the best for them for their particular disease at that time point. So we do have to deal with that issue.
The other aspect is, and this was brought up yesterday, CARs are used, and I think rightfully so, as a bridge to a bone marrow transplant. So allogeneic transplant is what many of these patients wish for, and they wish they could qualify or have their cells their state of disease be in a remission, so that they will be selected for an allo transplant. And the CARs, the expectation that is to get to the transplant of the patient. So the CARs is just another, round of treatment that will get them to the transplant, just like those other cycles of chemo were, several months ago. So that's a level of expectation that's a little bit different. They're looking at the CARs to get them to where they are. Imagine they needed to get to when they started down the treatment for their ALL.
One thing I would let the audience know is we're down to like the last 13 minutes. So if anyone has a question, feel free to raise your hand, and then we'll get a microphone to you. While we're waiting for a microphone, I would say, do you have any experience with patients who relapse after a CAR T cell therapy and then going on to another CAR T cell trial?
I don't.
I haven't. We've used PD-1 inhibitors after CAR T cells on trials. They didn't seem to work. But we haven't used CAR T cells sequentially.
It's very
We've used the same
for
patients who had a remission that didn't last forever. We retreated them with CAR T cells to get them into a second remission, and that occasionally works, and then take them to transplant.
Okay. Yes.
Thanks very much for all of this. It's been super interesting. I'm wondering if you could sort of put some numbers to the conversation. I'm interested in how many commercial patients you've actually treated with CAR T, and also if you could provide some insight about the patient experience. It's my understanding that patients generally are required to be within a mile of the hospital for 30 days with the caregiver, and that the SOP is generally inpatient for 7 to 9 days. Is that about right in your experience?
To answer that first. So at our institution with a commercial product, we've treated about 20 patients at this point. We do have these rules in terms of proximity. They have to be within, we say, half an hour of the hospital, 8 miles, not 1 mile, and not in Mexico, because that's in that region. They have to have a caregiver 24/7 for the first at least 2 to 3 months, we say. They're not allowed to drive for the first 8 weeks after they get the CAR T cells or operate heavy machinery, which is a problem in California because that's how people get around. And a lot of, so we try to find housing for them near the hospital, which it's not always easy, and it might be a financial burden for them. And they have to find the caregiver.
So yeah, we in LA, it's in terms of, we don't talk about mileage, we talk about minutes on the road. So 60 minutes is what we allow them to be away from the hospital. Of course, that changes from time to time during the day, but that's what we do. And we also have facilities for patients to stay, at least for that first week, on the grounds after discharge. So that's, and we've treated, I actually don't know how many patients we've treated, but it's quite a few.
How often, or do you know how often like a patient is unable to fulfill like these, I guess, non-medical, more social criteria, and therefore aren't able to be treated with CAR T cells?
I'd say about 10% of patients might not be able to fulfill that.
Yeah, we go through, we have the committee that approves the patient has certain criteria, one of which is they have a good caretaking facility. Now, if they don't have a caretaker, we actually will provide a caretaker if they will agree with that. But also they go through a psychosocial evaluation to make sure that they understand the issues involved, that they're committed to this, it's going to be a long-term process that they're motivated for, and that sort of thing. So in addition to getting authorization and to fulfill the label, we call it, that is, if they meet the label for the commercial product, that's the first critical hurdle that the committee looks at to decide whether the patient can go forward.
Are there any other questions from the audience?
Sure. Matthew Durdy from Cell and Gene Therapy Catapult. I'm intrigued by the comment about the $1.5 million worth of hospital costs. And we're familiar with the cost of the active substance and the drivers of that cost, but what are the elements of that hospital cost that make it so much greater? Is it the complexity? Is it the risk? Is it the unfamiliarity with the use of the product? And are there things that we can do in future generations of the products to work that out of the system so that they become easier to use or cheaper to use?
Well, the patients are very sick. If you can use the CAR T cells when they're not sick, that might be a major solution. Use it up front. Give them a little induction chemotherapy, and then give them CAR T cells. I mean, is that in the works?
Yeah, I mean, if we use them up front, they won't be as sick. But also, if there are better ways to prevent or treat CRS and neurotoxicity, we will be able to do this on an outpatient basis. Or even if there were some biomarkers to predict when they're going to develop these toxicities, we could, because right now we have to keep everyone in the hospital for at least a week. Like I mentioned before, we have to have an ICU bed available for them, which is also a big cost. And it's impossible to predict who's going to develop these toxicities because there are patients who might only have very mild CRS that could have been treated outpatient.
One more question.
Any other audience questions?
Can you guys hear me back here? Over here? I'm just wondering, for the indications where there are both products have been approved commercially right now, how do you decide which one the patient should get?
Again, it's the label that is looked at first, so that the age may be important. That might help there. I can't answer that question specifically. I think that's a...
So, it's a very good question, and we don't have a good answer for it. We have these two products. I have to say that our clinical trials were with Kite, so we had more experience with Kite, using their product and their process. So we've been using Kite almost exclusively for our lymphoma patients because of our experience with that. And when something is working, because we haven't had any issues with manufacturing or wait times or getting slots, so we're afraid to change our process, although it'd be more fair to split our patients in half.
Do you have, as a pediatric oncologist, I've always dealt with younger age groups, but do you have and with the commercial product for B-cell precursor ALL is generally for pediatric and young adults. Do you have any experience using the commercial product in older adults?
We don't. As a matter of fact, I just saw like a month ago, I saw in clinic a patient who with refractory ALL who had just turned 26 the week before I saw him. And I talked to the company, I talked to everyone I could to convince them that he should get CAR T cells because it's not fair. It sounds like we're discriminating against 26-year-olds when we only allow 25-year-olds to be on the trial. But I wasn't able to give him the CAR T cells. A very strict criteria.
It's kind of arbitrary, but still a line somewhere.
Yes, and for this unfortunate young person, it was like a week before he came to see me.
And I think one of the things that comes up as we get to the end of the session is like moving forward, where do things move? And there's always a lot of discussion on CAR T cell therapies in the clinic for solid tumors. And I didn't know if you had clinical trials at your institutions that you would be able to have any experience about some of the difficulties, challenges that offers compared to the hematologic malignancies that we've talked about the majority of this session.
Can you refine that question again? Could you repeat parts of that question again?
As we go forward, a lot of people are looking at solid tumor as a target. Yeah. And since we've talked mainly about hematologic malignancies, what are the unique aspects of using CAR T cells for solid tumors? And do you all have any experience at your institution in a clinical trial setting for that indication?
Yeah, we have quite a bit of experience. I mean, our first experience was with glioblastoma, of course, where glioblastoma results was actually very promising. And then patients began to relapse with this is an IL-13 receptor alpha 2 CAR T cell, and the tumor expresses IL-13 on the surface. So these T cells could find the tumor that way. And there was some remarkable case. There was a famous case that got presented at the New England Journal with a patient that had tumor all the way down his spine and in his brain. So rather than give the tumor, the T cells directly into the tumor as the protocol was written, the FDA allowed us to do a single patient where we injected into the ventricular space. And lo and behold, all of the tumor disappeared within the next week or two. And that patient went on to a very active life for about another 9 months and then relapsed with a IL-13 receptor alpha 2 negative tumor. And so we now have a HER2 protocol that is used with glioblastoma as well. And it's also used in brain tumor that's breast cancer metastatic to the brain. So those areas of solid tumor are where we're treating patients now. And there's a prostate cancer project that's moving towards the clinic as well. I think there's an ovarian cancer trial that's coming along.
I can tell you what issues we had at our institution. We do have clinical trials. The practical issues that we faced was that the BMT doctors who are used to cellular therapies and managing these complications, we have no idea about solid tumors. I mean, I don't even know glioblastoma. I think it's in the brain somewhere, but I don't know much more about it. And the solid tumor doctors who know how to treat these cancers, they don't know how to administer cellular therapies and deal with the side effects that we see after transplant. So nobody really wanted to do it, or everybody wanted to do it, I don't know. But the way we solved it was that we created a separate cellular therapy service for these patients, because I think that's going to be used much more frequently. I mean, these results are impressive for the solid tumors. So we have this service that's going to grow with input from both specialties.
You actually make a good point. It's very kind of unique in that you've got one set of oncologists has the patient and the patient disease experience, and the other set has the experience in administering the treatment.
Yeah, exactly.
So it's always a challenge at some hospitals, depending on how compartmentalized or siloed their clinical services
we have
are.
found, the hematologists have found, much to their surprise, that the medical oncologists are educable. And so they go through the same training, and we then certify a medical oncologist to treat certain.
Well, I think we've reached the end of our session, and I think it's been actually an excellent session. I thank the audience for their questions, and feel free to ask our panelists any questions in the break that follows.